Hello dear students, welcome in this channel Patel Science Academy. In today's lecture you are going to learn about the development of the endosperm. What is endosperm? Actually the triploid PEN primary endosperm Ik nucleus. It goes for a repeated cell division, mitotic type of cell division in which the number of chromosome remains same to form the nutritive structure, and that nutritive structure is called endosperm. Let's see it one more time. The PEN, which is triploid in number, this is PEN primary endosperm Ik nucleus. It divides and redivides by the process of the mitosis cell division to form the nutritive structure, and that nutritive structure is called endosperm. This is that endosperm. How does this 3N comes? I am explaining it right now. Actually I have explained it in my previous lecture too, when I have uploaded double fertilization, that lecture. So during the fertilization, as fertilization takes place two time, first fertilization in between the first male gamete and the egg to form the zygote, which is two in number, diploid in number, and second fertilization it takes place in between the second male, which is second male gamete, which is single N in and 2N secondary nucleus to give the 3N structure. So this is first N from the male second male gamete, and this is secondary nucleus of the carpel to form this 3N structure, which is triploid, and this triploid structure is called primary endosperm nucleus, that is PEN. So likewise there is a formation of this PEN by the process of the second fertilization in the plant. So this PEN, it goes repeatedly for the cell division, mitotic type of cell division. By this mitosis cell division process, it undergoes into so many cell division, and finally it forms a structure called this endosperm. So today in this lecture you are going to know about the development of this endosperm in this lecture. So in the post-fertilization, the changes within the ovule, embryo, and endosperm, all these are seen simultaneously, means at the same time. In the structure of the carpel, the ovule is there, embryo is there, and the endosperm is there. So development takes place in these all three structures simultaneously. The other cells of the embryo sac disorganize sooner or later. Means only ovule, embryo, and endosperm remains for the further development. The formation of the triploid endosperm nucleus, it triggers the cell division. Which cell division? Mitotic cell division, which leads to the formation of this endosperm, that I have explained diagrammatically too in the previous slides. So likewise there is a formation of the endosperm, and how does this endosperm develops, and what are the different types of the endosperm in the different plants of the world. On the basis of the mode of development, there are three types of endosperm. Number one, nuclear endosperm. Number two, cellular endosperm. And number three, alluvial endosperm. So one by one we'll see all these three types of endosperm. So here first of all we'll see this nuclear type of endosperm. What is this nuclear type of endosperm? This is actually the most common type of endosperm. It is found in about 161 angiospermic plant families, and primary endosperm nucleus repeatedly divides mitotically, but there is no wall formation at starting at beginning. This is the speciality of this endosperm. Nuclear primary endospermic nucleus divides, but there is no wall formation at the beginning, and it produces large number of free nuclei, and because of this large number of free nuclei are produced without cell wall, this is this endosperm is given name nuclear type. Big central vacuoles. Then what happens after the formation of this free nuclei? The big central vacuoles appears at the center and center of the cell, and it pushes all these free nuclei towards the periphery. And then what happens? Later the wall develops in between the nuclei, and hence this is the multicellular endosperm. So likewise there is a formation of this endospermic nucleus. So let's see it one by one. So this is that PEN. It goes for the free mitotic cell division, and it forms the so many small nuclei. So many nuclei are formed there by the mitotic cell division, but there is no cell wall formation. Thereafter what happens? A vacuoles is developed at center. You can see this vacuoles here. So say vacuoles develops, and this vacuoles pushes all the nucleus towards the periphery. You can see here all the nucleus goes towards the periphery in this structure. So what happens? At last the cell wall formation around the nucleus. So each nucleus eventually get protected by the cell wall. So cell wall is formed there, but at last. So likewise this type of endosperm, in which nucleus divides first, many nuclei, and then vacuoles develop at the center, pushes all the nuclei towards the periphery, and finally a cell wall is formed around each nuclei. Uh, this type of endosperm is called free endosperm, and this free endosperm is found in some examples I'm putting here. These are some examples of endospermic nuclear endosperm. Actually, in several plants cell wall formation remain incomplete. For example, in the wheat, for example in the sunflower, for example in the coconut. These are the examples of uh, nuclear endosperm in which the formation of the cell wall remain incomplete. So in the coconut, the multicellular endosperm in the outer part, and free nuclear as well as the vacuolated endosperm in the center is developed. So these are the example of uh, this free nuclei. Now we'll go for the second structure, and second type is, as you know, this is second type is cellular type of endosperm. So what happens during this cellular type of endosperm? uh, in some plants, the division of the triploid PEN nucleus is immediately followed by the wall formation. So here the difference uh, lies in between the cellular and the nuclear uh, type of endosperm. Here endosperm PEN is mitotically divide, and it is followed by a quick formation of the cell wall, so that the endosperm is cellular right from the beginning. Means cell wall is formed right from the beginning around the each nuclei. This is the difference in between this nuclear and the cellular type of endosperm. So these are mostly observed in 72 families of the dicotyledons, and diagrammatically it is likewise. You can see here diagrammatically it is likewise. So you can see here this is that PEN, primary endospermic nucleus. It goes for the cell division and divides into two nuclei, and you can see here each nucleus is now having this cell wall, which is start uh, from beginning, and around this cell wall there is a formation, around this nuclei there is a formation of the cell wall. So uh, likewise this is the formation of the nuclear of the cellular endosperms, and then eventually cellular endosperm right from the beginning. You can see here different nucleus around protected or surrounded by the cell wall is formed right from the beginning. So this type of endosperm is called cellular endosperm. So in this cellular endosperm you can see a few example of this cellular endosperm out of this 72 families of dicotyledons. So it is found in the balsam plant. It is also found in this petunia plant, and it is also found in the adoxia plant. this is balsam plant, petunia plant, and adoxa plant. So this is about the um, cellular type of this is about the cellular type of endosperm, the second type of the endosperm. Now we'll go for the third type of endosperm, and third type of endosperm, it includes the intermediate type, and it is halobial type. This is halobial type, and it is found in the monocotyledons order Halobialis. So that's why this term is used here, halobial type of endosperm. So this is third and last type of the endosperm. And what happens during this endosperm? The endosperm development, first division of the primary endospermic nucleus PEN is followed by the transverse wall, and which divides the cell unequally. So it means nucleus start developing here by the mitotic cell division, by going into the by repeatedly division, cell division, mitotic, and it goes thereafter for the transverse wall formation. Because of that transverse wall formation, there is not a equal division of the structure in that cell, and unequal cell are formed there. So there are two unequal cell formed: number one, the smaller one, and number two, the larger one. So a smaller one is called the Chesa cell, and the larger one is called micropyle cell. So there is a formation of the Chalaza cell and the micropylar cell. Then what happens? Then the nuclei in each cell divides by the free nuclear division. Then a wall develops between the nuclei in the micropylar chamber. So uh, in this chelazal cell and the micropylar cell, nuclear cell division takes place, but the cellular formation is found only in the micropylar cell around the nucleus, not in the chelazal cell. So then it is that's why both type of endosperm are found in this halobial structure, a nuclear too and cellular too. That's why it is called the intermediate between the cellular and the endosperm, nuclear type of endosperm. The example of this endosperm is aspodylus, and we'll see all this example diagrammatically. You can see there, this is the example of the this is the example of the alluvial endosperm in the aspodylus plant. And now we'll see it diagrammatically, what happens during this development of halobial endosperm. We'll see it now properly. So as you can see, this is PEN, primary endospermic nucleus. It goes for the first mitotic cell division, and which is followed by this transverse cell wall formation. So because of this transverse cell formation, because of the nuclear division, one nucleus divided into two nucleus. So this two nucleus is formed there, but there is a formation of this transverse wall, and because of this transverse wall, there are two cells are formed. One is larger, another one is smaller. The smaller one is called chalaza cell, and the larger one is called micropyle cell. So what happens with this smaller one and the larger one cells, which is having this nucleus? What happens with this cell? It goes for the free nuclear division, and the nucleus from this micropyle as well as from this chalaza cell divides and redivides and converts into many nuclei. Likewise here, this is the nucleus, these are the nucleus of the chalazal cell, and these are the nucleus of the micropylar cell. So nucleus are dividing, but thereafter what is happening? The nucleus in the region of this micropylar chamber, it develops, it is surrounded by the cell wall. You can see here, cell wall is developed in the region of the micropylar chamber, but there is no cell wall in this chalazal chamber. So the nucleus in this chalaza remain without cell wall, and this the micropylar chamber is having the nucleus surrounded by the uh, cell wall. So likewise this is the development of the uh, halobial endosperm, which is the intermediate endosperm in between the uh, nuclear type and the cellular type. So this is about this lecture, development of endosperm. In this lecture I have explained you about the three type of the endosperm, that is uh, nuclear endosperm, cellular endosperm, and the halobial endosperm. I hope you have understood all these three types of the endosperms, which is actually developed from the PEN, primary endosperm Ik nucleus, which is triploid in number. This is about this lecture. Thank you very much. This is Shahzad sir saying you goodbye. So please subscribe my channel, Patel Science Academy. Don't forget to subscribe my channel, share it, like it, and comments. If you are having any question or confusion in your mind, I will try to explain all that answers in a very easiest way. So this is Shahzad saying you goodbye, and keep healthy, stay at home, and subscribe my channel. Thank you very much.